Hello everyone, I am Miss Rakhi Shinde and today we are going to learn about the concept of gene regulation in prokaryotes, that is the Lac Operon. So let's start with some basic term which makes to easy understanding of a gene regulation. So first of all, what is the meaning of gene? Gene is the segment of DNA and that controls all traits of the organism, that may be a physical or metabolical. In prokaryotic cell, for example bacteria, the gene regulation mainly occurs at a transcription level because the prokaryotes or prokaryotic cell do not have a nucleus. So the transcription and translation occurs simultaneously in the cytoplasm. So the lifetime of a mRNA is very short. So therefore, the easiest and fastest way to control the gene regulation is by starting and stopping of a transcription. As we know that the genomic DNA of a bacteria has a many genes, and the genes by the process of transcription and translation forms a protein. It means that the information encoded in the DNA is transcribed into the mRNA through a process of transcription and then translated into a protein through a process of translation, and whole this process is known as the gene expression. The ability of cell to switch the gene on and off, it is a fundamental importance because it enables the cell to respond to a changing environment, and it is the basis of a cell differentiation. So we can say that the gene regulation is a process in which cell determine which gene it will express and when. So the gene regulation is used by a cell to increase or decrease the production of a specific gene product. The two scientists, that is Jacob and Monod, has discovered the gene regulation in prokaryotes, especially in a E. coli, and named as the Lac Operon. Lac Operon. The most of genes are regulated by the extracellular signal in case of the bacteria, and which are usually molecule in a growth medium. The signal can positively regulate the gene expression is known as a activator, and the signal can negatively regulate the gene expression is known as the repressor. The activator is also known as the positive regulator because it positively regulates the gene expression, and repressor is also known as the negative regulator because it negatively regulates a gene expression. And both the activator and repressor are the DNA binding proteins and regulates a gene expression. Okay, let's start with the operon. So first of all, what is the meaning of operon? So operon is a group of genes. Group of genes, or the genes are organized into a group, that is the operon. The operon can be a inducible and operon can be a repressible. Inducible operon is a catabolic process and repressible operon is the anabolic process. The Lac Operon is the example of inducible operon, and trip operon is the example of a repressible operon. So what is the difference between inducible operon and repressible operon? So the inducible operon is usually off and turn on when the inducer molecule like lactose binds to an inactivate the repressor and activating a gene expression for a catabolism. And repressible operon is a usually on and turn off when the co-repressor, for example tryptophan, the co-repressor tryptophan activate a repressor and inhibit a transcription for a anabolism. So it means that the inducible operon is associated with a catabolic process and repressible operon is associated with the anabolic process. Also it means that inducible operon is the catabolic process and activated in presence of a substances, and repressible operon is associated with the anabolic process and inhibited by the presence of a specific synthetic products. Then why the inducible operon is the catabolic and repressible operon is the anabolic? Inducible operon is the catabolic process because it controls a gene for breaking down of substances. For example, lactose. That lactose is breakdowns into the two components, that is glucose and then galactose. And repressible operon is associated with the anabolic process. Or why it is called as the anabolic process? Because the repressible protein is the anabolic process because it controls the biosynthetic pathways. For example, building blocks of a complexes. For example, amino acid, that is the tryptophan operon. So Lac Operon is the example of inducible operon and trip operon is the example of a repressible operon. Okay, let's see the structure of Lac Operon. So operon is made up of several components and each with a specific function. The operon consists of a promoter, operator as well as a structural gene. So what is a promoter? Promoter is a DNA sequence where the RNA polymerase can bind and start the transcription. Operator is also a DNA sequence nearby a promoter where the repressor protein can bind and blocks a transcription. And structural genes. The structural genes codes for an enzyme as well as a protein which is needed for a specific function. And there are three types of structural genes: LAC-Z, LAC-Y and LAC-A gene. And these structural genes which is transcribed as a single mRNA in a single promoter. So LAC-Z gene encodes the enzyme beta galactosidase. LAC-Y gene encodes the enzyme permease. And LAC-A gene encodes the enzyme transacetylase. Transacetylase. It means that this LAC-Z gene is responsible for the synthesis of beta galactosidase enzyme. LAC-Y gene is responsible for the synthesis of permease enzyme, and then LAC-A gene is responsible for the synthesis of a transacetylase enzyme. Next is that there is a one site, that is the CAP site, catabolic activator protein site, where the CAP protein can bind and increases the affinity of RNA polymerase. It means that this CAP protein play an important role in a turn on the operon. Turn on the operon. So this is the structure of a Lac Operon. Next is that the importance of a gene regulation. Why the gene regulation is important? So the gene regulation is important for a energy conservation, of a cell, cell differentiation, it is important for a growth and development of a cell, it is important for the efficient use of our resources, efficient use of our resources, then control of metabolic pathways, control of metabolic pathways. It is important for rapid response mechanism, rapid response, rapid response. Rapid response mechanism and survival and evolution. So, gene regulation is important for energy conservation, cell differentiation, growth and development, efficient use of our resources, control of metabolic pathways, rapid response mechanism, and survival and evolution. Next is that how the lactose transport into the cell and breakdowns into the two components, that is glucose and then galactose. So there is a one enzyme that is a permease. That permease enzyme transport a lactose into the cell or inside a cell. And inside a cell, there is a one enzyme that is the beta galactosidase. These beta galactosidase hydrolyze this lactose into the glucose as well as the galactose. Then this beta galactosidase also converts the lactose into a allolactose, and this allolactose is a actual inducer. This allolactose binds to a repressor and inactivates a repressor. It means that these allolactose play an important role in a turn on the operon. Turn on the operon. So in this way, the lactose enter inside the cell and breakdowns into the two components, that is glucose and galactose, and also some molecules of lactose is converted into the allolactose, and that allolactose play an important role in a turn on the operon. Okay. Next is that, so what turn on the Lac Operon? In a media containing both lactose as well as the glucose, but E. coli prefer a glucose over a lactose because glucose require a less energy to break down as compared to a lactose. But if lactose is only present in a media, it utilizes it as a energy source. So E. coli undergoes a Lac Operon. Only two conditions are favored. First one is the availability of a lactose, and second one is the glucose not available. So, to utilize a lactose as a carbon source, E. coli must express its operon gene for lactose uptake as well as the lactose metabolism. In E. coli, the Lac Operon works under the two levels of control. First one is the negative control, is known as the negative regulation. Positive control, that is the positive regulation. Negative control, it is controlled by a repressor protein, and positive control, it is controlled by a activator protein. So let's see, there is a two scenario, that is the presence of lactose or absence of lactose. So there is a one gene that is a regulatory gene, that is the LAC-I gene. That LAC-I gene forms a protein, that is the regulatory protein, that is the repressor protein, and In presence of in absence of a lactose, the repressor protein can active. If the repressor protein active, they can binds to a operator, and if once the repressor protein can binds to a operator, the RNA polymerase cannot binds to a promoter. It means that RNA polymerase cannot move forward. So there is a no transcription of any genes. It means that there is a no synthesis of the beta galactosidase enzyme, no synthesis of the permease, no synthesis of the transacetylase. It means that the phenomena should be a off, or operon should be a off. Next scenario is the presence of lactose. This regulatory gene or Lac I gene forms a repressor protein through a process of transcription and translation. But in presence of lactose, the some molecules of lactose will be converted into the allolactose. And as we know that this allolactose is the actual inducer. So in presence of lactose, the some molecules of lactose will be converted into the allolactose by using a enzyme beta galactosidase. And this allolactose binds a repressor and inactivate the repressor. If the inactivated repressor cannot binds to a operator, it cannot binds to a operator. So the RNA polymerase can binds to a promoter and start the transcription. It means that there is the synthesis of beta galactosidase enzyme, permease, as well as the transacetylase. Simply, if no lactose or absence of lactose, repressor protein or repressor active. If repressor active, decreases the affinity of RNA polymerase. Decreases the affinity of RNA polymerase. And operon should be a off. And in presence of lactose, if lactose is present, repressor protein or repressor inactive. If repressor protein is inactive, affinity of RNA polymerase is increases, and then transcription should be a on. So this is a negative regulation. Next is a positive regulation. As we know that the E. coli prefers a glucose over a lactose because the glucose gives a quick energy. It means that they are directly enters into the glycolysis. There is a no extra enzyme synthesis is needed. Efficient energy carbon source. So if the glucose is available, cell does not want to use a lactose, and this control is known as the catabolic repression. Catabolic repression. Catabolic repression is the suppression of a Lac Operon gene expression in presence of a glucose and resulting a reduced lactose metabolism. It means that glucose works indirectly through a cyclic AMP as well as the cap protein. Here, this glucose is inversely proportional to a cyclic AMP. The glucose level of glucose is high, the cyclic AMP is low. If the cyclic AMP is low, cap will be a inactive. If the glucose level is low, cyclic AMP is a high. And if the cyclic AMP is a high, cap will be a active. And cyclic AMP, that is the CAM, is a activator protein. Then there is also a two scenarios when the glucose level is a low and glucose level is a high. Glucose level is a high. So when the glucose level is a low, E. coli synthesize the cyclic AMP, and if the cyclic AMP is high, they can bind to a cap protein, and this complex binds to a DNA sequence or binds to a promoter. And as we know that this cap protein increases the affinity of a RNA polymerase, and transcription should be on. It means that there is the synthesis of some enzyme as well as the sugar molecule. Then if the glucose level is low, the glucose inhibited the synthesis of a cyclic AMP. If the cyclic AMP is inhibited, they cannot bind to a cap protein. Then cap protein will be inactive. So this cap protein cannot bind to a promoter, and there is a decrease the affinity of a RNA polymerase. So there is a no transcription as well as a basal level of a transcription. Simply the glucose level is low, CAMP is high. CAMP is high, CAP be active. If CAP active, increases the affinity of RNA polymerase, and transcription should be on, or operon should be on. And opposite to that, if the glucose level is high, cyclic AMP is low. Cyclic AMP is low, CAP will be inactive. If CAP will be inactive, the affinity of RNA polymerase is decreases, and transcription should be off. So this is the gene regulation in prokaryotes, especially in a Lac Operon. Okay, thank you.